Hello, my name is Ed Neilan. I'm the Chief Medical and Scientific Officer at NORD, the National Organization for Rare Disorders. Thank you for your attention. The title I've chosen for this presentation is RDCA-DAP, Patients' Contributions of Data and a Physician's Perspective. I'll spend most of my time talking about concrete contributions of data into the RDCA platform, but I wanted to put it in that slightly broader perspective as a practicing pediatrician and medical geneticist. NORD for 38 years has been a leading nonprofit organization that fights to improve the lives of patients with rare diseases. We do this by supporting patients and member organizations, accelerating research, providing education, disseminating information, and driving public policy. My disclosures. In addition to my role at NORD, I'm an attending physician in genetics and metabolism at Boston Children's Hospital, and I own stock in Sanofi Genzyme, where I previously worked. Here's a brief outline of my comments. I'm going to make a few remarks about what I see as the special value of patient-owned data. Let me be clear what I mean by that. I mean data gathered in a proper scientific way by a patient organization that has as its primary mission to make maximal best use of that data, such as when one of NORD's disease-focused member organizations undertakes to collect its own data. Now I'm going to talk a little bit about NORD's AM Rare Registry and Natural History Study platform, and then focus on NORD's role in coordinating transfers of patient-owned data into RDCA-DAP. I'll highlight specific data contributions, including making a focused case study on data contributions into RDCA-DAP from the registry organized by NORD for the Foundation for Prader-Willi Research. So, patient-owned data, as I defined it before, is likely also patient-informed data. So you have a group of patients and their advocates looking at the problems their patients are facing and trying to add information that may advance the field for their own benefit. Particularly in rare diseases, where there's often incomplete knowledge and limited clinical experience available in the medical literature, this is very important, as it may be difficult for physicians who've not personally taken care of patients with that disease to be sure what to include in clinical trials, and patient-informed data helps sometimes to define the most appropriate endpoints for clinical trials, which may need to be novel endpoints specific to that disease. Patient-owned data may also bring attention from biopharma companies, academics, and the FDA to rare diseases that might otherwise remain neglected. Of the 7,000 estimated rare diseases, more than 90% have no FDA-approved treatment, and currently the attention of the pharmaceutical industry is on 100 or 200 of the 7,000 diseases for the most part. So this is also very important. Finally, patient-owned data collected by patient groups whose driving interest is to advance their disease towards a treatment is less likely to get stuck in a silo where only one or very few organizations have access to it. NORD's AM Rare Registry and Natural History Study platform is a suite of custom-designed software that runs on a NORD server and is designed primarily to collect data through surveys that are online forms completed by patients or their caregivers, and allow the collection of demographic data as well as patient-reported natural history data. The platform has grown to also allow the capture of other kinds of data, and further additions will allow a more formal route for clinicians to contribute data about their patients as well. Most of the AM Rare registries are created for and owned by patient organizations who have chosen to work with NORD in order to find a platform to run their own data collection. Some of the initial NORD AM Rare Registry launches were supported by an FDA grant to NORD. Currently, there are 29 active AM Rare registries, a few that have essentially been retired, and many more that are in development. At present, there are nearly 12,000 consented participants in these registry and natural history studies, and more than 132,000 survey forms have been completed to contribute that data. NORD's role in the RDCA-DAP, which you've already heard described well by other speakers in the opening sessions, is multifaceted, but I will focus here strictly on NORD's role in transferring de-identified data sets from the AM Rare Registry platform into c-path for inclusion in RDCA-DAP. This is done only after a voluntary choice to share data is made by the organization owning the data, and after they're signing a negotiated data sharing agreement. The data is carefully de-identified and prepared into the proper format by NORD before it is transferred to c-path. This is a work-intensive process, which was done with a lot of direct involvement by NORD personnel. But we're looking forward to the implementation soon of a new API that was developed by NORD in a collaboration with c-path to efficiently accelerate that process. The future data sets will be processed by the API and then cross-checked by human effort, making the process more efficient. I don't think I'll spend too long on this slide as it is more general, but it does reflect some of the feedback received from c-path as they analyze the initial data contributions, as well as some general principles of what the content of optimal data contribution should be. As it says here, the components that are needed to ensure that data contributed to RDCA-DAP is robust and useful generally includes transferring the data dictionary, that is the metadata, explaining the meaning of each variable, standard and applied reference terminologies, survey instructions to help with understanding exactly what questions were answered, and the study protocol and manual of operations, as these core data elements can help researchers better understand the data that has been collected. Key data elements that should be transferred, if they can be, include population demographic information, diagnostic information, comorbidities, treatment and medication information, adverse events, and clinical outcomes. This slide lists NORD's completed data contributions to the RDCA-DAP at this time and the source registries, those for the National PKU Alliance, the International Pemphigus and Pemphigoid Foundation, the Foundation for Prader-Willi Research, the Spinal Muscular Atrophy with Respiratory Distress Type 1 patient organization, and registries for necrotizing enterocolitis and congenital hyperinsulinism. Plans have also been made and are progressing to submit data collected on additional rare diseases. Let me turn now to a little bit of an in-depth case study on one of those data contributions, dealing with Prader-Willi syndrome. Prader-Willi syndrome is a complex, contiguous gene syndrome resulting from loss of expression of certain imprinted genes located near each other in a particular region of chromosome 15. The major features of Prader-Willi syndrome include severe hypotonia and frequent feeding difficulties in infancy, followed later in childhood by developmental delays, and an ironic and serious reversal of the feeding problem, where patients develop severe hunger or hyperphagia and eat too much, gain weight, and suffer from complications of weight gain as well as related behavioral difficulties. The Foundation for Prader-Willi Research created a disease-specific registry on NORD's AM Rare Registry platform and have used it to conduct natural history research on multiple aspects of Prader-Willi syndrome. FPWR has managed and leveraged this data effectively, resulting in more than one scientific publication and others that are currently in the works, as well as additional summaries of data posted on their website, which help to highlight and better describe the extent of about a dozen features of Prader-Willi syndrome. The Prader-Willi Research Foundation has thus far chosen to make two separate sequential contributions of data sets from their NORD AM Rare-based registry into RDCA-DAP. FPWR's registry population includes about 2,000 patients with Prader-Willi syndrome, and the scope of the data collected involves approximately 65 different carefully designed survey forms. FPWR, advised by its Medical and Scientific Review Committee, initially chose to contribute a subset of their data that they had selected to include many features of possible interest for cross-disease research. That initial data submission, which has already been de-identified and prepared by NORD and also already received and processed by c-path for inclusion in RDCA-DAP, includes de-identified patient demographic information, information on age and weight, remembering here that extraordinary hunger and excess weight gain is among the primary concerns in the disorder, height, race, ethnicity, and sex. Diagnostic information including summary of genetic test results, general medical history, gastrointestinal health and feeding, again central to the condition, and neurological history related to the equally important feature of developmental delays and neurological issues in the syndrome. Subsequently, after having a good experience with the first data contribution, FPWR has decided to contribute a second set of data, including more than 4 years of data on approximately 600 patients, including feeding behavior changes and weight changes over time, and potentially related other behaviors such as nighttime awakening to seek food, as well as survey data outlining the parentally implemented behavior controls at home and changes over time in those control measures. Typically, these control measures include things like locks placed on refrigerators and food cabinets to prevent children with Prader-Willi syndrome from overeating when they aren't under direct supervision. This data was collected roughly every 6 months, and so it's multi-dimensional data around the critical eating behaviors in the disease. This data set will be prepared soon by NORD for transfer using the new API. FPWR hopes that the sophisticated data analysis capabilities of RDCA-DAP will be of use in correctly interpreting this complicated data, which has temporal relationships and potentially could identify causal relationships in this complex longitudinal data. Frankly, the Foundation feels it's beyond their ability to interpret the data fully. You can learn more about FPWR's experiences and their decisions to contribute data to RDCA-DAP when FPWR's Director of Research Programs, Dr. Teresa Strong, speaks this afternoon as part of a live panel discussion on successful importation of data into DAP, lessons learned from importing registry data into RDCA-DAP. Thank you very much for your attention.